Okay, all. Hopefully you guys, not too many of you have watched the video already. I actually re-recorded this video because I figured out something about why I'm having so much trouble with drawing. And it's actually the program that I'm using to record and not the Bamboo itself that I'm using. So I pre-drew out some of these pictures in hopes to shorten this lecture. So I'm going to dive right in here into changing the resting membrane potential. So now we've established that resting membrane potential, that electrochemical gradient, where we have a higher concentration of sodium outside, right, the neuron membrane, and then a higher concentration of potassium inside the cell. Also, remember that we have, again, the negatively charged ions lining up at the interior of the membrane, and then the positively charged ions at the exterior, okay? So this is what establishes that electrochemical gradient, and now we're going to use that electrochemical gradient to transmit these messages, okay? So again, membrane potential here is going to change when concentrations of ions across the membrane change, okay? And this is going to have to obviously be because there's some change in membrane permeability to these ions. And this is where those gated channels are going to come in handy, okay? So now let's talk about the two types of signals. We can have a graded potential, which is going to be a very localized change across the cell membrane. And it's going to typically happen in your dendrites as well as your cell body, not in the axon. And it's not propagated. And it's graded. So you can have a little bit of a change or you can have a big change. Okay. Now, action potentials are going to be different. They're going to work across the axon, the long distance of the axon. So again, here, if you want to put graded potentials are only seen in the dendrites and the cell body. So put dendrites and cell body here. Action potentials are going to be on the axons and they are going to travel the whole distance of the axon, okay? And the difference is they're not graded. So this is an all or none deal, and I know you probably have heard that before. So we're going to get into exactly what that is. So again, changes in membrane potential are going to be used to receive signals, to integrate those signals, and then eventually send that information down the axon, okay? So let's get into how this happens. So our graded potentials, again, are just these small deviations from our resting membrane potential. Remember, resting membrane potential is negative 70 millivolts, okay? So we can have kind of two different types of graded potentials. And again, this makes graded potentials different than action potentials, is we can go kind of two separate ways. So we can have a depolarizing graded potential, and we'll talk about this a little bit later in this lecture. This is called an excitatory potential. And sorry, so this is your depolarization. I should here draw a nice connection here for you. So your depolarization is actually going to be this one. I should have flip-flopped these. And your hyperpolarization is actually going to be this one, okay? So just so that you don't get confused, I just didn't realize that the diagram was flip-flopped there. So, oh, and it, of course, got rid of my drawing. But you saw, okay, so depolarization is going to be any time that we have a decrease in the membrane potential where we're moving up towards closer to zero, okay, closer towards the positive. So basically, in this case, the inside of the membrane is becoming less negative. And so if the inside of the membrane is becoming less negative, that means we're having an influx of positively charged ions. And as you can guess, because we've already established that sodium gradient, it's going to be sodium that's going to be rushing in, bringing in all of these positively charged ions, making that inside of the cell less negative. That's what we call depolarization. It's making the membrane literally less polar, okay? Now, in hyperpolarization, that's what you're seeing here. This is hyperpolarization, and these are going to be actually inhibitory membrane potentials, and you'll see why in just a minute when we talk about action potentials. Hyperpolarization is an increase in the membrane potential towards and making it even more negative. So we've been bringing it farther away from zero. So for example, you might get an inhibitory, neuron inhibitory synapse here where that neuron is signaling and releasing neurotransmitter and telling this area of the membrane to open up its potassium channels. If potassium opens up, now remember, potassium is in a higher concentration inside the cell. So it's going to move down its concentration gradient out. So you're going to have a bunch of positively charged neurons leaving this area, making it even more negative. Okay. So this is called hyperpolarization. Okay. And again, these two things are going to happen in our graded potentials. Hyperpolarization again is and depolarization, where it's just, again, it can be different varying levels. That's what we mean by graded, is going to occur in the dendrites in the cell body. Okay, if that makes sense. This might make a little more sense as we continue on here. So again, graded potentials, just as a review, and I'm going to review all of this at the end, but I'm kind of giving you the pieces to the puzzle, are going to be short-lived, they're localized, and again, they're graded. So the stronger the stimulus, the more of a change across the membrane there is, and the farther the current's going to flow. Okay. So meaning, again, if you have a stronger excitatory stimulus, it's going to cause, again, more of those sodium channels to open. These are going to be ligand gated. We'll, again, talk about that coming up here. And then, again, if we have that stimulus and it opens, again, these gated ion channels, that's where it's going to result in either depolarization or hyperpolarization. Okay, just like we talked about. Now, we can have what are called receptor potentials or generator potentials. And these, again, are going to be these potentials that are in receptors of your sensory neurons. So, for example, like, when you have those, when we talked about some of these things like Meissner's corpuscles and things like that. So, what can happen with some of these is they actually have like these remember those mechanically gated channels? And as you push on your skin, you're really causing those channels and those neurons and those sensory receptors to deform. And that is causing an influx of ions, causing those mechanically gated channels to open. And then we also have postsynaptic potential, where it's just, again, a neuron talking to another neuron. So again, we'll kind of get into all this as we go through this. Okay, and so this is just kind of showing you again what happens in depolarization here, which we're going to kind of talk the most about. And so again, if you get some type of stimulus, again, maybe it's a mechanical stimulus, and all of a sudden it pushes on these sodium gated, ion channels and allows sodium to move in, okay, to that interior that's, again, more negatively charged at the membrane, then that's going to cause a change in the membrane potential, making it more positive, okay? And then sometimes that depolarization can spread, and we'll talk about how it does that, kind of like, again, the waves, spreading out from a droplet. So we'll talk about how that happens. Okay, so again, this is just with our graded potentials. I kind of just talked about this, but when we talk about graded potentials, this is going to be very different than action potentials. Graded potentials are going to arise from either a mechanical stimulation or a chemical stimulation. So again, this is really important to know, okay? Mechanical stimulation or chemical stimulation. This is going to be different from our action potential, which is going to arise from a change in voltage and electrical stimulation, okay? So think graded potentials are either mechanical or chemical. And what that means is the mechanical stimulation is literally, again, putting pressure on those, causing those mechanically gated ion channels to open, okay? By physically putting pressure on them and physically opening them. And then chemical stimulation is going to be, of course, by releasing something like a neurotransmitter. And we saw this in the neuromuscular junctions, remember? We're releasing that neurotransmitter across that synaptic cleft, and then binding to this ligand-gated channel, so binding to a receptor on that channel and causing that channel to open, allowing something like sodium to rush into the membrane, okay? And I'm going to go through this whole thing in just a minute in a drawing. So again, please keep in mind that this is for graded potentials, and the amount of change, again, here can vary with the strength of the stimuli. So that's all very different than an action potential. And remember that this change is just going to be a local change. It's not going to spread throughout the whole neuron. It's very localized. And so let's talk about some of these in terms of excitatory, inhibitory. I know I get through this later on in the lecture, but I think it's kind of interesting. And then I'm going to kind of introduce action potentials here, and then we'll really get into the nitty-gritties of it. So I'm going to try to draw on this with my pen, but I realized again that it's really, for some reason, this program is really slow. So let me go ahead, and we're going to talk a little bit about what happens here in the dendrites in the cell body, and then how we can actually trigger an action potential. Now, remember that action potentials are going to be triggered here, okay? That is the axon hillock, where the cell body connects to the axon. And again, this area right before that kind of initial segment here is going to be called the trigger zone. That's where we trigger what's called an action potential. Okay? But in the dendrites and in the cell body here, we're only going to be triggering these graded potentials. So these two are the graded potentials. And when we do that again, we can have excitatory graded potentials that are triggered, or inhibitory graded potentials, depending upon, again, what neuron is synapsing here and what neurotransmitter is being released. So please keep in mind that there are many different neurons oftentimes that are synapsing with this one neuron, okay? And most of the time your excitatory neurons are actually going to be synapsing with the dendrites, even though it's not drawn here. Usually they're with the dendrites, and then the inhibitory ones are usually on the cell body. Not always, but again, for the most part. So now let's talk about what happens. Let's say that here you are, you're this neuron here, and you're getting all these signals from these different neurons, okay? Some excitatory. In this case, this neuron here is releasing some excitatory neurotransmitter across that cleft. It's binding to the receptors on the surface here, and as it binds to something like this sodium channel here, and it's only going to bind to the sodium channels because it's excitatory, it's going to cause that sodium channel to open up, okay? Now remember, sodium is going to want to move down its concentration gradient that was already established by that sodium-potassium pump, and it's also going to be attracted to the negatively charged interior from the resting membrane potential. So sodium is going to rush in, boom, okay, causing the interior of the cell membrane to become more positive, okay? But again, only in this area because it's localized graded potential, okay? Now, on the flip side of things, you can have these inhibitory ones like this guy over here, okay? And instead, this inhibitory one is releasing inhibitory neurotransmitter, and it's binding to maybe something like a chlorine channel that's going to allow, oh, I forgot that it was doing this before, I apologize. So it may do that to like something like a chlorine channel that might allow negatively charged chlorine ions in, or it can allow potassium out, because remember, we have that gradient established already where we have a higher concentration of potassium inside. So let's use that example. Let's say our neurotransmitter here binds here to our potassium channel, opening this potassium channel and allowing, excuse me, it's doing, let me turn this off. So allowing now potassium, right, to rush out. Now, as potassium rushes out, okay, we're having a bunch of positively charged ions leaving, and it's going to make the inside of this membrane more negative, okay? So now what you have here is you have some neurons making the inside of the membrane, okay, more positive, kind of bringing us closer to what we call this threshold point, okay? And then we have some other inhibitory neurons that are releasing neurotransmitter that's bringing us farther away from the threshold, okay? So what is threshold now? Let's talk about that. So remember that our resting membrane potential started at initially negative 70 millivolts, okay? I'm not going to draw the millivolts because this is so hard to write with. And our threshold here, this little dotted line, okay, is our all or none point for an action potential. And this is going to be negative 55, okay? And so what's going to happen here is the summation, we'll talk about this a little later too in the lecture, the summation of, again, all the positively charged particles depolarizing the membrane here and the negatively ones hyperpolarizing here is going to have to bring us up to negative 55 millivolts for an action potential to be generated at that axon hillock, at that trigger zone. So, okay, what happens is, let's say in this case, in this example, we have a lot more excitatory, okay, neurons releasing a lot more excitatory neurotransmitters and causing a lot more of those sodium channels to open up, more so than here, the inhibitory ones that are causing hyperpolarization. So if we get more depolarization than we do hyperpolarization, meaning we get more positively charged ions, more positively charged sodium ions coming in, bringing us closer to threshold, if eventually we change that membrane potential so much that we hit this negative 55 millivolt point right here, okay, then, and if that point is hit, if it changes so much in the interior of the cell body here, that at this trigger zone, okay, we hit that negative 55 millivolt point, then an action potential will be triggered, okay? And once that action potential is triggered, there are going to be these electrical gated channels in here. We call them voltage gated channels that are going to open when, again, we hit that negative 55 millivolt. It will trigger them to open. And once it does, there is no stopping it. Now, this one's going to trigger, this one's going to trigger, this one's going to trigger, and sodium is going to rush in, okay, causing the action potential to propagate all the way down to, again, the axon terminals and eventually the synaptic end bulbs, okay? And so that's what this is. This is an action potential. Again, when we hit that negative 55 millivolt change, we are triggering now here these electrical or voltage gated channels to open, okay? And all of them are going to open. So it is going all the way down the axon. We are hitting full action potential, okay? So let's talk a little bit more about what happens in this action potential, okay? So what I really wanted you to see back there with this was that graded potentials can lead to an action potential, okay? So keep that in mind, graded potentials can lead to an action potential. Now again, an action potential is an impulse that is going to travel all the way down the axon, okay? And it's going to fully reverse the membrane potential. And again, we call that depolarization. Now we do have to eventually, obviously, restore that resting membrane potential so that we can, of course, fire again, right? So we've got to restore that membrane potential back to resting state. So that way, then if we want to, we can, again, trigger another action potential, or maybe we don't want to, okay? So it stays at resting. So let's talk about this action potential and this all or none state that it's in, okay? So I'm going to go through step by step the action potential. Now, remember, now that we're talking about an action potential, this is only in the axon, okay, from the trigger zone all the way down. So the first step that's going to happen, obviously, is we're in resting state, okay? So when we're in resting state, we now, again, are focused on these voltage-gated channels. These are not triggered by neurotransmitter. They are not triggered by any mechanical stimulus. They are only triggered by a change in voltage to threshold, okay, through that negative 55 millivolts in the axon. Okay, so in these gated channels, if you look, the sodium gated channel is going to have an inactivation gate, okay, that they kind of draw hanging off here, and then activation gates, okay? So right now, the inactivation gate is actually open, which means basically that it can be triggered, okay, to be open. Now, if we'll talk about this in a minute. If the inactivation gate is closed here, not even a voltage, again, change up to negative 55 millivolts across the membrane can trigger this. So a short timer is going to be inactivated and completely closed, and we'll talk about why that is. But then we have also these activation gates, and these activation gates are the ones that are triggered by a change in the membrane potential. Now, potassium only has the activation gates, okay, and we'll talk about why this is. The other piece to this puzzle is that when a change happens across the membrane here, sodium channels are going to open rapidly. So the activation gates are going to open rapidly. Potassium channels, I want you to think about their activation gates like a creaky door. They're going to kind of slowly open. So what's going to happen is the sodium gated channels are going to open and close by the time these potassium channels finally open up, okay? So that's another piece of the puzzle to kind of keep in mind as we go through this. Okay, so again, first step is we're in resting state. Only the leakage channels for sodium and potassium are open. Remember, especially again, those potassium ones, letting potassium out are really helping to maintain our resting membrane potential. So the sodium channel again has the activation gates and the inactivation gates. And at rest, the inactivation gate's open, okay? And the activation gate is what's closed. And that activation gate will only open with depolarization, okay? So let's get to depolarization. All of a sudden now, again, we're in the trigger zone here of the axon. And all of a sudden there was enough of a change from all those graded potentials here that there was enough of a change, depolarizing, change in the membrane potential that it brought us up to that negative 55 millivolts. Boom, at that negative 55 millivolt point, all of a sudden, that change in voltage is going to trigger both the sodium and the potassium channels to open. But remember, again, potassium's really slow, so it's going to stay closed. So you can kind of think of it, if it's easier for you, that potassium is still closed. It is still closed because it's triggering so slowly, okay? So right now, depolarization, the sodium gated channels open really quickly, and it's only those activation gates. So the activation gates open really quickly because they were activated by the change in voltage. Sodium is going to rush in. As sodium rushes in, it's going to change this membrane potential, right? Making it more positive on the inside. And it's going to become so positive that's going to hit about plus 30 millivolts. So it's going to completely turn that membrane potential upside down, going from negative to more positive, okay? Until it's going to hit a peak at this certain point here, okay? Now, so again, this is just kind of basically what I told you, everything that happens, that threshold, negative 50 to 55 millivolts, we have that positive feedback mechanism that's going to occur. And all of our sodium channels in that area are going to open up, okay? Causing more of a change in the membrane potential. And you're going to see this is going to cause neighboring sodium channels to open up, causing more of a change in membrane potential. Hence, positive feedback. Okay, so that membrane polarity is going to jump about up to positive 30. Now what happens? Okay, so after we peak here, usually around positive 30 millivolts, what's going to happen is now the sodium channels are going to be inactivating. And so what's going to happen is the inactivation gate first is going to close. Okay. As it closes, finally, our potassium activation gates are finally open. And now potassium is going to exit. Now, as potassium exits, remember that, again, this is a positively charged ion leaving. It's going to reestablish our membrane potential, okay? So as we've got positive charges leaving, it's going to make the inside of the membrane more negative again. Boom, bringing us back to our resting membrane state, okay? So that's called repolarization. Again, sodium channels are inactivated by those inactivation gates closing, okay? Our action potential spike stops. Our voltage-gated potassium channels open and are finally open. and the potassium exits again outside the cell, reestablishing our membrane potential, and then we call that repolarization, okay? Then we have this fourth event, okay? And so what happens in this fourth event is that actually a little bit too much potassium leaves, okay? And this is actually a good thing because it's going to cause this slight refractory period where a neuron, it'll take a hyperstimulus to stimulate stimulate another action potential here. And that's because some of those potassium channels are just going to remain open and allow excess potassium out. And so actually it'll let so much out that our membrane potential will drop actually below 70 millivolts, okay? Up until, again, these potassium channels finally close, and then our resting membrane potential is reestablished by those sodium-potassium pumps, okay? So again, once all of this is done, what reestablishes from hyperpolarization here, what reestablishes our resting membrane potential is those sodium-potassium ATP pumps, okay, that are always going to be running. And so at this point, finally then, as only a few sodium channels are remaining open, but allowing hyperpolarization, finally here, the sodium channels are going to reset. So the inactivation gate's going to pop off or pop open, and now the activation gates are going to close. And so what that means is now this sodium channel can be stimulated by another threshold stimulus, another voltage change to negative 55 millivolts. So it's ready to go, okay? Now, in the hyperpolarization phase, you're going to see it's going to take an extra, strong stimulus, okay, to do that. So usually, again, this neuron isn't re-stimulated until we hit resting membrane potential. And this gives us a slight break in, our nerve transmission. So, and we'll talk about this. This is called a refractory period, okay, in just a minute. So again, this is just talking about the same thing, that hyperpolarization, all of the things that I just talked about. Now, again, keep in mind that after repolarization, the sodium-potassium pumps are going to restore those ionic conditions, restore our membrane potential back to where, again, it's at negative 70 millivolts, more negative on the inside, more positive on the outside. And it's also going to reestablish our chemical gradient with more sodium on the outside, more potassium on the inside, okay? So the sodium, potassium, ATP, if you think about it, this is why we need a lot of ATP, right? You have to constantly, every time you're doing nerve conduction, you have to reestablish this sodium-potassium gradient. And to do that, you have to do it with ATP because you're pumping, again, sodium out. And I know you can't see those sodium-potassium pumps, but if you could picture, they're off here, on the side as well, pumping 3 sodium out and 2 potassium back in. And since they're pumping across that concentration gradient, that's a lot of ATP. So anytime, again, your nerves are sending, again, this information and depolarization is happening, there's a lot of ATP that you're using to reestablish that concentration gradient. You can also see that without ATP, your neurons can't fire because you can't establish that sodium-potassium gradient. So again, ATP, really important for brain function, obviously. So this is a nice chart that goes over the action potential, but to be honest, I actually like this one better. So if you're going to print one and put it up on your mirror, which I highly suggest that you do because you brush your teeth every day, right? You can look at this. This is a great one because it goes over all of the steps. Here in resting state, again, all of the voltage-gated sodium and potassium channels are closed. And I love it because it shows the activation gate and the inactivation gate here, okay? Then, again, we get some type of depolarizing, again, signal. We hit that negative 55 millivolt point, or that change across the membrane, and we've hit threshold, and now our voltage-gated sodium channel is triggered to open, okay? Remember, it's going to open the fastest. And so first, that's our activation gates. That's what can be triggered, right? Sodium's going to flow in, okay, causing, again, an influx of positively charged ions. It's going to depolarize the membrane, opening even more sodium channel activation gates, okay, especially the neighboring ones. Then we're going to hit a peak, okay, where eventually now finally our inactivation gate is going to close. And once our inactivation gate closes on those sodium channels, we hit our peak for our action potential. Now, finally, the potassium channels are open. And so remember, they're the creaky door. And so finally, potassium can rush out. And as potassium rushes out, we can reestablish our resting membrane potential. Okay, that's our repolarization. Okay, bringing us back towards resting membrane potential. Then again, we have a little bit of excess, okay, the potassium that's going to leave. We're going to have a slight hyperpolarization phase, okay, where it's actually going to drop below resting membrane potential. And at this point, finally our sodium channels are going to kind of reset themselves. The inactivation gate is going to open, and now the activation gates are going to close. Okay, and that's going to return us to a resting state. Eventually that potassium channel will close too, and we'll return to this resting state where now again it can be activated. Okay, now keep in mind that during this phase right here, where that sodium inactivation gate is closed, this sodium channel is inactivated. It cannot open. So this is going to be what we actually call an absolute refractory period, because that inactivation gate cannot be triggered to open with any change, no matter how much of a change in voltage across that membrane. It's temporarily inactive, okay? And it cannot, this channel cannot be triggered again until that inactivation gate opens, okay? And then the activation gate closes, and then the activation gate's ready to be triggered again, okay? So I'm going to go ahead and draw this all out for you, just because I think that it's an easy way to kind of picture what's happening. And I pre-drew this all out for you so you can see this. So again, let's put everything together. Let's do big picture. So remember, we've got these graded potentials from, again, the synapses of what we call the presynaptic neuron. There again, before the synaptic cleft here. And some of these are inhibitory and some of these are excitatory, okay? So here, let's say we get, again, a signal from this excitatory one. Boom, okay? It's going to cause an influx here, or it's going to release neurotransmitter that's excitatory. This is called an excitatory postsynaptic potential. It's going to release neurotransmitter. That neurotransmitter is going to, I drew a really big synaptic cleft, is going to move across the synaptic cleft. Oh, man, come on. It's going to move across that synaptic cleft, and it's going to bind to the receptor on that sodium channel because it's excitatory, right? So as it binds, here, let me get it off that. As it binds, it's going to trigger this sodium channel to open. Now, remember, this is a graded potential. So again, it's going to be localized. And these are either going to be, again, ligand gated or chemical gated channels only, not voltage ones. So here, this is going to trigger an influx of sodium, okay? Now, again, that influx of sodium isn't going to trigger anything in here, okay? We need, again, more excitatory neurons, okay, to release more excitatory neurotransmitter causing more sodium channels to open. Sorry, I don't know why it keeps doing that there. So as it does that again, we're going to get that influx of sodium, right? Causing again a depolarizing effect on the membrane as we make it more positive. Now we do have some inhibitory, so we might be getting a few signals from inhibitory ones as well. And those inhibitory ones, again, might be releasing neurotransmitter here, and binding, that neurotransmitter might be binding to something like a potassium channel, allowing potassium to flow out, okay? And that's causing a hyperpolarizing effect, making it, again, more negatively charged on the inside, okay? So whether or not we get an action potential is all going to depend on summation of, so the addition of all of these action potentials together. And if you have more action potentials here that are excitatory than we do inhibitory, it can cause, again, the inside of the membrane to become more negative. So again, you know, it's, again, just adding up all of these potentials together, these graded potentials. So let's say in this case that we have a lot more excitatory potentials and excitatory neurons than we do inhibitory ones. And it's going to cause such a change, such an influx of sodium. Let me get my pen here again. Such an influx of sodium in the cell body here that it's going to allow, sorry, this is supposed to be red to show you my influx of sodium. So it's going to cause so much of an influx of sodium that it's going to cause, again, us to end up with a change right here at this trigger zone that's going to hit negative 55 millivolts. So again, all of our graded potentials here, okay, added up together, still brought our membrane potential up from negative 70 millivolts all the way up to negative 55 millivolts. What happens when we hit that negative 55 millivolt threshold right here at the trigger zone is that we've now triggered our voltage-gated sodium channels to open. So boom, here, this voltage-gated sodium channel is triggered to open, allowing sodium to rush in, causing depolarization of the membrane across here, okay? Now, as it does, it's going to trigger neighboring sodium-gated channels to open, okay? Propagating this message. And so this is going to cause an influx of, again, sodium into the cell, causing depolarization, causing these neighboring sodium channels to open, these neighboring voltage-gated sodium channels to open. So you can see how it's going to propagate, right, in this one direction, okay? So now, why doesn't the action potential move backwards, okay? So you know this. It's because eventually what happens is as that sodium-gated channel, again, starts to close, it's the inactivation gate that's going to close first. And as that inactivation gate closes, remember, no matter what happens to the membrane potential here, this gate is going to stay closed, okay? So what we've done here is, if I can draw this out, okay, right here at this point here, right, we've already hit the top of our action potential. And now this sodium-gated inactivation gate is going to close. And finally, our potassium channel is going to open. As that potassium channel, that voltage-gated potassium channel opens, potassium is going to rush out, returning the membrane potential back to its resting state, okay? Now, again, that potassium channel is going to stay open for a little bit too long, and it's going to cause a hyperpolarizing effect where we're actually going to drop below that negative 70 millivolt point, okay? And as that happens again, it's going to take what we call a super threshold stimulus to be able to stimulate that sodium-gated channel to open. So let me show you what I mean by this, okay? So hopefully that kind of helps put everything together. I also wanted to show you how this propagates down the entire neuron. So the reason that an action potential will propagate once you've hit, again, that threshold, and the reason it's all or none is that this sodium-gated channel is going to allow an influx, right, of sodium causing a depolarizing effect, and that then is going to stimulate neighboring sodium channels to open. So this one here, and then this one's going to cause a bunch of sodium to flux in, now causing a depolarizing effect and causing this sodium-gated channel to open. And then that one's going to cause this one to open here, okay? Oops, if I can draw here. And then, oh, it's doing this again. Sorry guys, this is actually, I found out the actual program that I'm using to record these. So my screen capture program. So anyway, as you can see, it's going to continue to cause these neighboring sodium channels to open, right? And that's why we get the propagation of this message all the way. I'm just going to go ahead and exit this and show you this. It's going to propagate that message all the way down the axon. Sorry about that. It's just really frustrating. Okay, so let me show you this on this actual graph here, which will kind of put it all together and really help it make sense. Okay, so here we're resting membrane potential, right? Right here, voltage-gated sodium channels are closed, voltage-gated potassium channels are closed. Okay, now if we get enough of a change, again, because of all those graded potentials, the addition of all those graded potentials, cause enough of a change at that trigger zone in the axon, we hit that negative 55 millivolt, because again, we're getting enough of a depolarizing change, right, with those graded potentials. If we hit that negative 55 millivolt point, boom, we've triggered an action potential at that trigger zone in the axon. Now we've triggered those sodium-gated ion channels to open, okay? And as they open, sodium is going to rush in, causing a depolarizing effect on the membrane, right? And this is an all or none deal, okay? So it's going to either trigger them open at negative 55 millivolts, or if it's a little under, then it's not going to trigger. If it's a little over, it's been already triggered. So it's not going to make any difference. It's still going to depolarize to the exact same point of about plus 30 millivolts. Okay. So what you're going to see here is, again, now our sodium gated channels in this phase, the sodium gated activation channels are open. The inactivation channels are open up to this point. Sodium's rushing in, causing depolarization up to about positive 30 millivolts. Okay. Totally flipping the membrane potential. Okay. Now, at this point here, we're going to start the repolarization phase. And that's when our sodium-gated inactivation gates are closing. So that's happening right here. Okay. And now our potassium channels are finally open. And as they open again, they're going to let that potassium out of the cell. Okay. Reestablishing our membrane potential. Okay. Again, causing that repolarization of the neuron, okay, of the axon there. Now, we call this phase right here absolute refractory, okay? And the absolute refractory, I like to think of the absolute refractory as the time frame where that activation gate cannot be triggered to open. So here in the green, in the depolarizing phase, it's already open. So it doesn't need to be triggered again. It can't be triggered again, right? Because it's already open. But here in this kind of beige area here, it also can't be triggered to be open, because that inactivation gate, no matter what type of stimulus you get, no matter what type of change you get in membrane potential, that inactivation gate will not open. And this is our absolute refractory period. What this means is you absolutely cannot trigger that sodium channel to reopen. Absolute refractory, okay? Now, as that inactivation gate opens up and then the activation gate closes on those sodium channels, now that sodium channel can be triggered to open again. But we're going to have what's called the relative refractory period, because look what happens here. Those the potassium channels, remember, are staying open a little too long, and they let lots of potassium out, and it actually brings us below resting membrane potential. So in this absolute refractory period here, what this means is we need a greater than threshold stimulus. So normally we just have to go from negative 70 to negative 55 millivolts. But here you might have to go from something like maybe negative 90 millivolts all the way to negative 55 millivolts. So we need a super strong stimulus, what we call a super threshold stimulus, okay, to bring us up to at negative 55 millivolts for that activation gate on the sodium channels to open. Okay, so that's why we call this the relative refractory period. So hopefully that helps make sense of the absolute refractory period and the relative refractory period. So I'm not going to play this for you just because I want to save time, but please take the time to play these flicks on generating an action potential. This will actually go open up my actual, and what I actually may do is I am going to do this. I am going to save just these slides that have the Pearson AMP flicks for you and post them so that you can open that and just watch these flicks. Please, please, please watch these. These will put everything together for you, okay? I'm not going to play them here because I found out in my last recording that it actually does something goofy, and it kind of, I don't know why. It doesn't sound that way to me, but it plays it like twice. And so it's really hard to listen to. So I will put that out there for you guys. Now, just remember again that action potentials are all or none. I like to think of it as kind of the analogy of, firing a gun. No matter how much, if you squeeze that trigger harder, it's not going to make the bullet go faster. Okay. You just have to squeeze the trigger to the point that it causes, again, the gun to fire. And again, it's not going to make that bullet go any faster. You just have to hit that point on the trigger that it actually causes the gun to fire. And now it's an all or none deal, right? There's no stopping. Once you hit that point on the trigger that the gun fires, there's no stopping that bullet either, okay? Just the same thing with an action potential. Once an action potential has been triggered, there's no stopping it, okay? So again, for an axon to fire, depolarization has to reach that threshold voltage change of about negative 55 millivolts. Now, once an action potential, we've already talked about this, is initiated, it's self-propagating, just like the wave. So once you start the wave again, just like, on a stadium, what's going to happen is, again, you can picture, here's sodium-gated channels, triggering more sodium-gated channels to open, triggering more sodium-gated channels to open, okay? So again, as those sodium-gated channels open, causing a depolarizing effect, it's going to cause their neighboring sodium channels to open. And thankfully, again, that wave moves only in one direction because of the sodium inactivation gates. They keep it from moving backwards. So that's why those sodium inactivation gates are so important. They keep the action potential from moving backwards, okay? So the action potential will only travel in one direction. So again, the propagation is just the term that we use for the action potential being transmitted all the way down the axon, okay? And again, that's because the sodium influx causes, again, depolarization in the neighboring areas, causing the neighboring sodium voltage-gated channels to open, okay? Pretty basic. And so this is just, again, a diagram kind of showing you that propagation of the action potential. Okay. And again, I have another flick on this. Please watch this. It's a really great flick. I love the Pearson flicks. All action potentials are alike. And again, are going to be independent of stimulus intensity, which means what that means is all action potentials are going to take you up to about negative 30 millivolts and then back down. Okay. No matter what. So, the way that the central nervous system actually tells the difference between a weak stimulus versus a strong one is actually the frequency of impulses or the frequency of action potentials. So again, if you receive, let me find one where I show you this. I have it here. So for example, again, here would be frequency. Like here, if you have a stronger stimulus, then you're going to have action potentials, again, that are firing as fast as they can, as fast as, again, those inactivation gates can open, and then the activation gates close back up, and then they're able to be retriggered, okay? So again, a higher frequency of action potentials means that neuron's firing as fast as it can, okay, all the way down that axon. Where, again, if you have maybe less of a stimulus, okay, then it's, the action potential might fire and then have a little break and then fire again and then have a little break. So that's what's going to make the difference in terms of, or that's how our central nervous system is going to be able to tell the difference between a strong stimulus versus a weak one. So again, higher frequencies, okay, action potential means a stronger stimulus. This is just kind of already what I talked about. It's just in the slide here for you if you like the verbiage on absolute refractory periods due to the inactivation of those inactivation sodium gates. And then the relative refractory is just where we need the super threshold, okay, stimulus. So I know I talked a lot about that, but I put all this, I like to put all this verbiage out there because you guys don't have a text to read. So you can read this like you can a text. Kind of fun, local anesthetics that you use, things like lidocaine, even certain neurotoxins, actually prevent the opening of voltage-gated channels. And so again, this would be important for especially our pain, our neurons that are taking information back about pain, right? If you're having something like a mole removed, you really are happy to not have those voltage-gated sodium channels working on your pain receptors and those sensory neurons in that area. because again, you don't really want to feel that. So that's what happens is basically, again, they're just preventing those voltage gated sodium channels from being able to open temporarily, right? Just temporarily. So the nerve impulse cannot pass that region there, okay, where we kind of shut down those voltage gated sodium channels. So that's how Novocaine and lidocaine work. Now, what's interesting is, and you guys that are in dental hygiene can correct me on this, but I believe that lidocaine is mostly used now instead of Novocaine for dental work. But you guys can correct me. Don't quote me on that because I'm not 100% positive on that. I just had read something on it. Okay. Continuous versus saltatory conduction. So remember that when we're talking about conduction down an axon, so we've only talked about unmyelinated fibers, okay? And so we talked about the continuous conduction all the way down, again, that axon. This kind of step-by-step, like the sodium channel here is going to stimulate, again, an influx causing its neighboring sodium channel right next to it to open, okay? And that can only happen in an unmyelinated fiber. Now, in myelinated fibers, where remember we've got the myelin sheath that's wrapped around the axon in certain areas, there are no voltage gated ion channels in that area that has the myelin wrapped around it. The only voltage gated channels that we're going to find in the nodes of Ranvier. And so what we see here is, so you may say, well, how does that work then? How do we even get nerve conduction down there? And believe it or not, it's actually faster because what happens is we end up getting depolarization only at the nodes of Ranvier. And in this case, what's going to happen is at these nodes of Ranvier, we're going to have a really high density, higher than the density of them all along the unmyelinated fibers. So you're going to have a really high density of voltage gated ion channels. And so when you trigger those voltage gated ion channels to open, it's going to trigger a lot of sodium to rush in. So much sodium is going to rush in that it's going to actually flow past that myelinated segment to the next node of Ranvier. Okay. And what it's going to do is it's going to cause depolarization to jump from node of Ranvier to node of Ranvier to node of Ranvier. And this is actually faster because you don't have to wait for so many voltage gated channels to open all along the entire axon. It allows it to jump. So this is a great diagram of this. Again, remember we have a higher concentration of these sodium gated channels. So what's going to happen is, again, as we hit threshold, the sodium gated channels are triggered to open. Sodium is going to rush in. There's such a high density of these sodium gated channels here that it's going to cause, again, so much of a change that it will actually hit threshold all the way down here. So again, that change in current is going to flow all the way past this myelinated region down here because so much sodium is rushing in. So again, as that happens, it's going to cause, again, these sodium gated channels now to open as they hit threshold. And literally you're going to see this is going to propagate this depolarization down the axon by jumping from node of Ranvier to node of Ranvier to node of Ranvier. And this is actually faster. So saltatory conduction is actually faster than continuous conduction, if you can picture, because you have to wait for this one to open, this one, and this one, and this one, and this one to open. Here, we're just jumping from here to here to here to here. So it's a lot faster. Speed of impulse propagation is, so not only is it going to be impacted by myelination. So now we know that myelinated fibers are going to conduct impulses faster because of that saltatory conduction, because they can jump. But also, obviously, the larger the fiber, the more surface area there's going to be for these, again, these sodium voltage gated channels. So it's also going to make nerve impulse conduction faster. Okay. So we have these three different types of fibers. So it's important that you kind of know these fibers, and we'll talk about where they are. So fiber A, the A fibers are going to be the largest ones, as you can see here, and they're also myelinated. So these ones are going to conduct the fastest. Okay, so the largest and the myelinated are going to conduct the fastest. So this is like your myelinated somatic sensory. So again, these are going to be kind of your body sensory fibers that are bringing information like touch information back from your skin. And then also your motor to your skeletal muscle. So that makes sense, right? Because this is what allows you to do such fast, fine motor skills. So very quickly, this information is being sent back. You detect the touch of your pen in your fingers, sends that information very quickly back to the brain through these A fibers, and then back out through these motor A fibers to fine tune your skeletal muscle actions in your hands. Okay. So it makes sense why these would be A fibers, right? You need that to happen fast. B fibers are kind of your like medium speed ones. These ones here are medium in diameter, as you can see here, and they're going to be medium in terms of speed. So again, they're myelinated. This is what we call our visceral sensory fibers. So these are the ones that are picking up sensory changes in your organs. So for example, again, as your bladder fills and stretches, okay, it's going to send information back through this visceral sensory fiber. And again, that's going to be a B fiber. Also, some of your autonomic, we haven't gotten into much of your autonomic ones, but some of your autonomic, what we call preganglionic neurons that are coming in are going to be that. So we'll get into that a little bit later when we get into the autonomic nervous system. And then our C fibers are going to be the tiniest ones, and they're unmyelinated. So these are going to be the slowest ones. And your unmyelinated ones are going to be your sensory, you have some unmyelinated sensory, not a lot of them, some of them are associated with special senses and things. And then you have the autonomic motor ones, which are, again, the ones that you don't think about, like your fight or flight motor and things like that. Okay. So our last slide here that we're going to talk about today is encoding of stimulus intensity. So how do we differentiate a light touch from a firm touch? And we already kind of talked about this. So one of the main ways that we can do this is obviously the frequency of impulses. So firm pressure will, again, push on more of those mechanically gated ion channels, causing more of a change in gradient potential and more of those neurons, right, to fire. So firm pressure is going to generate impulses at a much higher frequency, okay? And so not only are we activating more of the sensory neurons with that firm pressure in that area, but we're also going to be re-stimulating those same neurons that we're stimulating with a higher frequency, okay? And so higher frequency of impulse and the more sensory neurons that are activated, the more basically sensory information we're sending back to the brain that we're getting a firm touch, okay? So a light touch would be the opposite. Light touch, again, is going to have a lower frequency of impulses in each of the neurons that we're stimulating, as well as simply fewer of those sensory neurons are activated. So that's how, again, our central nervous system decodes the difference between firm touch and light touch. I'm going to go ahead and stop here, and then I will start up the next lecture here, just because this has already run pretty long. And as always, guys, if you have any questions, please feel free to ask me. I know this is some tough stuff, so please reach out to me.